Okay, so this is part 1 of chapter 9, microbial genetics. Make sure that you watch the second lecture video for chapter 9, part 2, which will be about change in microbial genetics. But for this first part, we're going to talk about the blueprint of life from DNA to proteins. So, the central dogma of molecular biology is the flow of information from DNA to RNA to protein. DNA is transcribed into RNA, and then RNA is translated into protein. And we'll talk more about the details of that. So let's zoom out first to talk about what is your DNA. Well, all the DNA that makes up an individual is called its genome, the complete set of genetic information within a cell. All right, and this includes chromosomes and plasmids, and they're composed of DNA. A chromosome in bacteria, so for E. coli, the chromosome is 4.7 times 10 log base E to the 6 base pairs long. The average size of a bacterial gene is about 1,000 base pairs. And E. coli has about 3,500 genes. Humans, in contrast, have about 30,000. Okay, so here you can see a size comparison. Here's an entire bacterial cell. This structure down here is the chromosome, so all this squiggly stuff. And over here, this single little loop, this is a plasmid. All right, so plasmids have genes for their own replication. They're independent from the bacterial chromosome. They may confer new characteristics to the cell, things like antibiotic resistance, toxin production, and virulence are all traits that are associated with plasmids. A gene is a functional unit of the genome. They code for a product. So genes are transcribed to RNA and then translated to a protein. Some genes are only transcribed to RNA. Not every A, T, G, or C in the genome codes for a gene product. So genes are special parts of the genome that actually code for protein products. DNA stands for deoxyribonucleic acid, and it's made up of nucleotides. Phosphate attached to deoxyribose, or pentose sugar. The sugar phosphate backbone builds up the sides of the ladder of DNA. And attached to that backbone are the nitrogenous bases, either A's, T's, G's, or C's. These nitrogenous bases are joined by covalent bonds between a 5' phosphate and a 3' hydroxyl. These are what hold them to the sugars and phosphates that make up the backbone or the sides of the DNA ladder. So if you picture DNA as a ladder, the sides of the ladder are made up of phosphate and sugar, and the rungs of the ladder that you step on, those are made up of the nitrogenous bases, the A's, T's, G's, and C's. The 5' and 3' parts of DNA are important because they help us know which way to read the code. So in DNA, we always go 5' to 3'. And these designations refer to the number of the carbon atoms on the pentose sugar of the nucleotide. All it is a way of orienting yourself when you're looking at a DNA molecule. All right, so DNA has a special structure in which it's anti-parallel. So the two strands of DNA are not only complementary, but also anti-parallel. So they're oriented in opposite directions. So you can see here, if we look at the red strand, zoom in, 5' to 3'. Oops. It goes 5' to 3' this way. And all of the oxygens are at the top points of those sugar molecules. If you look at the blue side that's opposite that, you see we've got phosphate sugar backbone on the outside again. Nucleotides are all in the middle. But this time, the oxygens are pointing down on the sugars. This is what we mean by anti-parallel. Okay, so we've got the phosphate sugar backbones on the outside here in the blue, phosphate sugar backbone on the outside on the right, and our base pairs of DNA in the center. And these base pairs are held together by hydrogen bonds. So we see A and T always bind together, and they share 2 hydrogen bonds based on those 2 little dots. And G and C always pair together. They share 3 hydrogen bonds. A mnemonic device you can use to help remember this is apples and trees, A and T go together, and cars and garage. All right, so the base pairing only happens in these very predictable ways. So DNA is double-stranded. It's helical, so that's that twisted ladder shape, and it has complementary base pairing. So adenine, A, only binds with thymine, T, and they have 2 hydrogen bonds that hold them together. Guanine has 3 hydrogen bonds and can only bond to cytosine. All right, so let's compare this to RNA. RNA is similar to DNA in many ways, but there are some exceptions. RNA is made out of ribose sugar in the backbone rather than deoxyribose sugar in DNA. RNA uses the nitrogenous base uracil instead of thymine. So if you see U's in a sequence, you know you're looking at an RNA sequence. RNA is usually single-stranded, so it doesn't have a complementary strand, and it's linear, not helical. RNA is usually found in shorter strands as well, because RNA generally codes for a single gene at a time. There are 3 functional groups of RNA. There are messenger RNAs, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, tRNA. rRNA and tRNA are not translated into protein. They're the ultimate products. Each are involved in protein synthesis. In bacterial DNA replication, before a cell divides, the DNA must be replicated, creating a second DNA molecule. Each cell after the division will have the same genetic information, and the offspring are an exact clone of the parent cell. DNA replication is generally bidirectional. Replication proceeds in opposite directions, and it allows for the entire chromosome to be replicated in half the time. Remember, bacterial chromosomes are circular. So we have these 2 replication forks where replication is working in opposite directions. Replication is semi-conservative, and that means that each of the new DNA molecules will contain one of the original strands. So this is the original DNA here in dark blue. You'll see that there's replication where each new strand gets one of the originals and then makes a complement to match it. If that first cell divides again, so the original cell divides to make 2 daughter cells. If those 2 daughter cells divide, then we see that the replication is semi-conservative again. So we still have the original blue copies Some cells will get one of the teal ones, and there will have to be some new replication of these lighter teal colors. But there's always one copy that's already made that's used as a template to make the complementary strand. All right, and it always happens in a 5' to 3' direction. So new nucleotides are added in 5' to 3' order. So this is the direction of synthesis. We're adding DNA down here because we're moving from 5' to 3'. So we would call this the new strand that's being assembled in the 5' to 3' direction. Above that is the template strand, and it's going to be going in the opposite way, right? 3' to 5' relative to this one. The enzyme that helps attach these new nucleotides is DNA polymerase. This is an important enzyme you'll need to know about. DNA replication requires coordinated action of many different enzymes and other proteins. DNA polymerases, and you know it's an enzyme because it ends with ASE, right? Are enzymes that synthesize new DNA strands. One strand is used as a template, and nucleotides are added one at a time. They always are added to the 3' end, and the new strand elongates in the 5' to 3' direction. Bacterial DNA replication is a process that's very accurate. There's really only one mistake out of every 10 billion nucleotides. DNA polymerase has a proofreading mechanism, so it can check for mistakes and correct them. Bacterial replication, the process. So specific proteins recognize a region on the chromosome, and that region is called the origin of replication. Helicase, another enzyme, right, ASE, helicase separates the double-stranded molecule. So helicase pulls apart the helix. Primase synthesizes small fragments of RNA called primers. Another enzyme, right? Primase. Good. So here we see helicase separates the double-stranded DNA molecule. Primase synthesizes an RNA primer, a short strand of RNA, to attach to the template strand. Synthesis continually proceeds of one of the new strands in the 5' to 3' direction, and this is called the leading strand. So steps 4 and 5 occur at the same time. So from those primers, DNA polymerase adds the new bases and moves on in the 5' to 3' direction to build a complementary strand and the leading strand. On the lagging strand, DNA polymerase can't add nucleotides to the 5' end. So the synthesis has to be reinitiated or restarted periodically as the helicase unwinds the DNA. That's called the lagging strand. A different DNA polymerase removes the RNA primers and adds DNA nucleotides. All right, and step 7, DNA ligase seals the gaps between the fragments on the lagging strand. So ligase, this enzyme, links the fragments together on the lagging strand, and so it's one long string of DNA. These fragments are called Okazaki fragments. All right, good. So here we've got our original DNA. DNA helicase separates the double-stranded DNA molecule. Primase synthesizes an RNA primer, which binds to the DNA template. Polymerase adds nucleotides in the 3' end of the fragment. So up here, this strand that can build continuously, this is the leading strand. And then down here, we have replication happening in short chunks. These fragments are called Okazaki fragments, and this is the lagging strand. Synthesis of the lagging strand is discontinuous. Synthesis is reinitiated periodically, generating a series of fragments that are later joined. Gene expression. So now moving on from the structure of DNA, let's talk about how it's used. Gene expression can be defined as the production of a protein from a gene sequence. And gene expression involves two separate processes, transcription and translation. Transcription, or making RNA, DNA is used as the template, and most genes, or DNA, code for proteins. The genes need to be transcribed into messenger RNA, or mRNA, and the mRNA is then translated into a protein during protein synthesis. So here we have double-stranded DNA. What will happen is this will unzip so that mRNA can access the gene in the DNA. And the mRNA will build a complementary copy. So you can see where they match up here. A, but the T's are replaced with U's, C, A, and so on. And this process from DNA to making a complementary mRNA strand is called transcription. So transcription begins when RNA polymerase recognizes a nucleotide sequence on DNA called the promoter. And then elongation can happen when RNA polymerase initiates synthesis without a primer. Finally, termination. Transcription ends when an RNA polymerase encounters a terminator sequence. So transcription begins with initiation, elongation as the RNA molecule is created, and then termination when it's at the end of the RNA. Translation happens when the mRNA is used to create a protein. So if we think of, let's go back here. If DNA is like the cookbook, right? mRNA might be a copy of a single recipe from the cookbook. And then we need to take that cookbook to the kitchen. So the kitchen, in this case, is the ribosome. So the mRNA travels to the ribosome, and that's where the proteins are made. That's where the recipe gets made. All right, so if DNA is the cookbook, mRNA is a copy of a single recipe. It needs to go to the ribosome, to the kitchen, and that's where the proteins are made, or the recipe or the meal is made. All right. mRNA is a temporary copy of genetic information, and the mRNA code is deciphered using the genetic code, which correlates to 3 sets of nucleotides. So there's 3-letter nucleotides which function as codons, and each codon codes for a single amino acid. So you can think of every 3 letters of DNA or mRNA as coding for an amino acid. All right, there are 64 different codons. 61 translate to the 20 different amino acid. There's 20 possible different amino acids, right? And more than one codon can code for a specific amino acid. All right, so here's a good codon chart. So the letters here, we see U's, so we know we're looking at mRNA, not DNA, right? So if I gave you a codon, say the codon was UAC, you'd go first letter U, second letter A, third letter C. Here it is. So the 3-letter mRNA codon UAC codes for the amino acid tyrosine. And if you study this chart, you'll notice some things. Down here in green, there's a methionine, a start codon. Proteins start with a methionine. So we can do a search through a DNA sequence or mRNA sequence for AUG to find where genes begin. And they all end with, a stop codon. There are 3 different stop codons, but same thing. All genes end with a stop codon, which is awfully convenient. All right, translation happens in the ribosome. So this is the site of protein synthesis. The ribosome is the kitchen. It's made of protein and rRNA. And ribosomes bring each amino acid into a favorable position so that an enzyme can catalyze a peptide bond. tRNA are segments of RNA that carry specific amino acids, and they act as a key that interprets the genetic code. The recognition is possible since the tRNA has an anticodon. So it's complementary to the mRNA. Let's look at an example. So we've got, all right, here's an amino acid up here, aspartate. It's held on by a covalent bond. So here's our tRNA, and it's got an anticodon down here. So if the mRNA says GAC, then tRNA knows it needs to go bring CUG because that will bind with AUG. Does that make sense? And if you look at it a little bit more zoomed out, the anticodon's down here. Here's the structure, and here's where it carries the appropriate amino acid that goes with this codon. All right, regulation of bacterial gene expression. Control mechanisms due to changes in the environment. So an energy efficient strategy would be to control the synthesis or the making of enzymes so you only make what's required. And to do this, cells control the expression of certain genes. And this makes sense, right? Like if we've got 3,000 different genes in a bacteria can't possibly be making 3,000 different proteins at once. It would be way too costly. It would use too many resources, too much energy, and it's not necessary. And by the same token, your cells aren't making 30,000 different gene products all at once. We only make what we need when we need it. All right, and a good example of this is the lac operon model. The lac operon consists of 3 genes involved with lactose degradation in bacteria. And the effect is that the genes are only expressed when lactose is present, but glucose is absent. So a very specific set of circumstances. And they use quorum sensing. So bacteria can sense the density of cells within their own population. Bacteria use chemical words to communicate, and it activates genes that are only beneficial when expressed in a large mass of cell. Cooperative activities which form biofilms are controlled by this. Bacteria hold off on attacking the body until suitable numbers are present to go against the immune system. So pathogens will wait until their numbers are sufficient before they begin to actually cause illness, which is pretty crazy. All right, the lac operon, we already did that, wrong way. So let's talk about an example. Staphylococcus aureus is a gram-positive coccus bacteria. It's responsible for skin infections, boils and pimples, surgical wounds. It's generally treated with methicillin, which is a penicillin-like antibiotic, since the 1970s. Now, however, 60% of Staph aureus strains are isolated, are resistant to methicillin. So methicillin is no longer effective in treating most Staph aureus infections. Vancomycin has been considered the drug of last resort, and even that is now showing to be ineffective. In 2002, a diabetic foot ulcer was isolated a strain of vancomycin resistant Staphylococcus aureus in Detroit. And the organism was also resistant to other common antibiotics like penicillin, methicillin, and ciprofloxacin. Here's a picture of the ulcer. All right, so that wraps up Chapter 9, Part 2, our discussion of DNA and transcription and translation to produce proteins. Make sure that you watch the second Chapter 9 lecture video on genetic change in microbes.